You're at the cutting edge of cardiovascular sciences. I'm John Cooke, professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist. And today we're going to have a very special guest who's going to be telling us about metabolism and the heart. And my co-host, Dr. Li Lai, who's an assistant professor here in the Department of Cardiovascular Sciences, will be introducing Dr. Rong Tian.
Good morning, everyone. Today is my truly privilege to introduce today's distinguished seminar speaker, Dr. Rong Tian. Dr. Tian is a leader in cardiac metabolism research and the professor and the director of the Interdisciplinary Mitochondria and Metabolism Center at the University of Washington. Dr. Tian got her MD from West China University of Medical Sciences and obtained her PhD from Aarhus University in Denmark. Following that, she did her postdoc at Harvard Medical School. Dr. Tian is internationally recognized by her work in bioenergetics, metabolism, and mitochondrial biology, which has significantly advanced our understanding of how metabolic pathways are rewired in the heart under stress. Her early studies were among the first to demonstrate how cardiac remodeling involves a shift in glucose and fatty acid metabolism. Beyond cardiac metabolism, Dr. Tian also made major contribution to the field through her work on NAD metabolism, mitochondrial stress responses, and inflammation. These discoveries have inspired new directions in the development of mitochondria-targeted therapies in cardiovascular disease. Currently, her lab is delving into the mechanistic role of mitochondria in cell-cell communication and their contribution to the development of heart failure with preserved ejection fraction, or HFpEF, a topic continues to pose significantly clinical challenges. In addition to her outstanding scientific achievement, Dr. Tian also served as the editor-in-chief of Journal of Molecular and Cellular Cardiology, further helping to shape the future of cardiovascular sciences in a global scale. Please join me welcome Dr. Tian.
Thank you, Dr. Tian, for joining us.
Thank you so much for having me, and also thank you for that flattering introduction. I'm so honored.
I ran into one of our faculty members today in the hallway, Dr. Dale Hamilton. He's very interested in your talk today, and so will others. There will be calls coming in during the show, and I would like to let the folks know at home how they can call in. If you're on your phone, you can just text to 37607, text DeBakey to that number. That's 37607, and then just text in your message. We'll get that, and then we can ask Dr. Tian your question. Or if you're at your computer screen, go to pollev.com. That's POLLEV.com. Enter DeBakey and ask your question. So, Dr. Tian, we're very interested in vascular.
I educated in China and received my MD. There, and I had an opportunity to go for a PhD study in Europe, and I went to Denmark, Aarhus. That was in the 80s, where for Chinese, very few people even know where Denmark is, and so it's quite an adventure, and it was an amazing experience. I think it really changed my life and how I look at the research. So after I studied, and at the time I studied angiotensin II inhibition of ACE in treatment of heart failure, and so I was into heart failure research. And when I was looking for a postdoc studies bioenergetics of the heart, and I learned that the heart is the organ that consumes most energy, and the energy metabolism must have played a role in the developing heart failure. And that was a time that metabolism is not a hot topic, and most people think everything about metabolism and the TCA cycle is already understood, and so the focus is more on the gene expressions, the oncogenes, and that was the exciting finding at the time. And now, several decades later, we find out that oncogene product actually regulate metabolism. So we're back to the metabolism topic, but at a much higher level now with the new methods and technologies, we can much better understand energy metabolism. So I feel very lucky that I'm on this topic, and it is a good time to study metabolism.
Mm-hmm. Wonderful. Well, with that introduction, maybe we'll go ahead and with your first slide and get started on the talk. And Dr. Tian has said that she would be happy to take questions during her talk, so please again, either text DeBakey to 37607 or go to polev.com and enter DeBakey and ask your question. Okay, Dr. Tian.
All right. Can you see the slide all right?
Yes, perfect.
Okay. Thanks everybody for dialing in. And the topic today is about mitochondria in heart failure. So I want to start by giving a very brief introduction about mitochondria so we can all get on the same page. Mitochondria is present in nearly all eukaryotic cells except red blood cells. It's a very unique organelle in a way. I'm trying to get a pointer here. So it's a Hey, can you see a pointer on the screen?
Yes, we can.
Okay, that's wonderful. Okay, so this is a very unique organelle. It has double membranes. It's a very sophisticated build and consists over 1,000 proteins. The special things about mitochondria is this double membrane structure makes the interior of the mitochondria impermeable to outside, and that basically is prevent the internal content of mitochondria get out to the cytosol, at the same time allow it to maintain a membrane potential. So it's almost like a small cell inside the cell. And another special feature about mitochondria is it's arrived from an endosymbiotic event. And that means this mitochondria has a reminiscent component from a bacterial origin. That's maybe one of the reasons mitochondrial content are not supposed to get into the cytosol. Mitochondria has a large number of functions. I have trouble to advance the slides. Here, okay. Most well-known function is its role in bioenergetics. It's considered a powerhouse of the cell. It produces ATP, regulate redox state. But the mitochondria also plays a very important role in biosynthesis for cell growth and proliferation. This is especially important for development such as stem cells and also in cancer. Mitochondria is not just an organelle to produce things, but it also regulates a lot of signaling pathways so that the cells can maintain a homeostasis in terms of function and metabolism. With all that in mind, and for the heart, mitochondria is critical for energy supply. So the cardiac contraction never stops. But the cardiac myocyte has a very low storage of ATP. So this ATP consumption and production has to be maintained on a B2B balance. And this is why the cardiac myocyte has the highest abundance of mitochondrial volume, and it consumes the highest amount of oxygen per unit weight. And so the human heart actually is going to turn over many times its own weight of ATP during every day on a daily basis. So with this knowledge in the background, you probably want to ask, if the heart fails, does the energy system fails as well? And very interesting thing, and probably also a bit challenging, is that we can't measure mitochondrial function in a living heart in a human being. The closest we can do is to measure the energy compound levels in the heart using the non-invasive MRI scanning in combination with spectroscopy. So shown here are a normal heart, a dilated cardiomyopathy heart, human heart. So with imaging-guided spectroscopy here, you can see the high energy phosphate content in a normal heart, a very nice peak of phosphocreatine and ATPs, and the ratio is about 2. And in a very severe dilated cardiomyopathy, stage 4 heart failure, this ratio dropped to below 1. And what's really important is that if you look at the risk ratio and you follow the patient in the long term, the ratio actually tracks with the long-term mortality. And at the time when this paper was published several decades ago, and this is a better predictor of mortality compared to ejection fraction. So this result itself doesn't say it's the energy deficiency that kills the heart. It's just a relationship or biomarker, if you will. But this raises the question whether the mitochondria is impaired in the failing heart. And since we can't measure directly in a live heart, we can at least take a look in the heart tissues from explanted heart or biopsies. So here is a heart tissue from a non-failing heart, and the heart failure with reduced ejection fraction due to decompensation to pressure overload. And you can see the swollen mitochondria with this array of the crystals. And the couple of decades later, and the biopsies from non-failing heart and the heart failure with preserved ejection fraction, we see almost a similar pathology change. And note that here with diabetes, but no heart failure, we don't see mitochondria abnormalities at the EM pictures, suggesting the mitochondrial impairment is really specific for the heart failure status. And now, there are many studies looking how the mitochondrial dysfunction would exacerbate the heart failure progression. But there's also a question of how mitochondria become dysfunctional, because this heart failure patient doesn't start with a mitochondrial defect. It starts with some other stress, but it impaired mitochondrial function at this stage. So there's a lot of study looking at how the mitochondria is made and how the mitochondria is maintained. As I mentioned before, mitochondria is an organelle encoded by 2 genomes, the nuclear genome and the mitochondrial genome, which is considered a reminiscent of bacterial genome. And there's already studies showing that the downregulation of PGC1 alpha, a master regulator of mitochondrial biogenesis in the failing heart, but targeting PGC1 alpha alone is not effective to rescue the heart failure. And then this question raised whether the mitochondrial DNA has also contributed. So a little bit about mitochondrial DNA here. Mitochondrial DNA is a very small circular DNA structure. Its transcription is different from nuclear DNA transcription. So it makes a transcript around and resulting in a long string of polycistrinic transcript. And this transcript has to be post-processed by cutting out the tRNAs in the middle, those yellow beads, and by our RNase-P function. And so Matthew Walker, who was, when he was a postdoc in the lab, discovered something very interesting in the human failing tissue. He found that in the failing tissue, there are some partially processed transcript. There were 2 mRNAs are still connected, and these accumulate in the heart failure tissue. And he was able to reproduce this finding using a mouse failing heart model by pressure overload tag. And you see all these pink bars that shows accumulation of a partially processed mitochondrial DNA transcript. If you map these transcript, there are always two mRNAs connected by a tRNA, so suggesting there's a defect in RNase P function in the tRNA excision. And very intriguingly, when we treat animals with NR, which is a precursor of NAD, we use this treatment to manipulate NADH redox state, we're able to restore the function. We're able to restore the transcript processing, and we no longer see accumulation of partially processed transcript. So why does that happen? It partially due to the RNase P subunit 2, MRPP2, which is NAD/NADH dependent short-chain dehydrogenase reductase. And so the function of this is dependent on NAD/NADH redox state. And Matthew's study showed indeed MRPP2 function is decreased in the failing heart. It can be restored when we increase NAD levels, and it's substrate azocarnitine or hydroxyazocarnitine also accumulate in the failing heart. Restored MRPP2 function can restore the levels of these compounds. As you know, if we increase NAD level, we'll change the NAD-dependent protein deacetylase function, such as sirtuins. And in the failing heart model, we use TAG model. You could see that TAG will induce protein hyperacetylation. And then if we provide NAD precursor, we increase NAD level, and this hyperacetylation will normalize, and summarized here. To see if this is responsible for what we see of improvement of MRPP2 function, Matthew introduced SIRT3 knockout animal, which is hyperacetylated across the board, is not responding to NAD levels because of lack of SIRT3, the deacetylase. But in a wild type animal and SIRT3 knockout, the induction of heart failure will reduce the NAD/NADH ratio, and this can be restored in both genotypes by providing more NAD. So this is also associated with improvement of deacetylase, dehydrogenase function of our MRPP2. So we consider the improvement of MRPP2 function is sensitive to NAD/NADH redox state rather than just the protein acetylation effect. Here, the data shows that if you normalize MRPP2 function, you would normalize the transcript, the gene expression that encoded by mitochondrial genome here, that there's a downregulation in heart failure and normalization by treatment. It doesn't really affect the genes encoded by nuclear genome. Either it's not unregulated or it's not responding to the treatment. Similarly, functionally, looking at the mitochondrial enzyme activities such as complex 1 and complex 4, which has a significant component encoded by mitochondrial genome, it's very responsive to the treatment. And the citrate synthase, which is also a mitochondrial matrix protein, it's encoded by nuclear genome, and it's not responsive to the treatment, suggesting this is, again, it's a mitochondrial DNA-coded protein effect. And we see the same for the SIRT3 knockout animal as wild-type animals. So it turned out that there's a large family of NADH redox-sensitive short-chain dehydrogenase reductase in the heart. We call it SDR. And the MRPP2 is just one of them. And our RNA-seq results show there's about 40 SDRs expressed in the heart. And the single largest group of SDR that contributes to one metabolic pathway is the retinol metabolism pathway. And these SDRs are responsible to turning into the inactive retinol into active retinoic acid. So we did mass spec analysis in collaboration with School of Pharmacy, looking for upstream metabolites of this pathway, such as retinoic esters or retinols, and see a significant accumulation in the failing heart, and the treatment can bring it down significantly. And if you line up the measurement with multiple groups and look at where they stand in redox state versus the amount of inactive compound retinol, and there's a beautiful correlation here suggesting this, again, this metabolism is dependent on NADH redox state. So at the same time, we realized in the literature, this Hopkins group has already discovered that the retinoic acid level in the failing heart, including failing human heart, is decreased. And they used all-trans retinoic acid to treat their animal models of heart failure and showed very significant improvement, but they did not report the mechanism of this function. So we went back and did bioinformatics analysis from differentially expressed genes after treatment, and the result shows multiple transcriptional pathways could be responsible for the benefit of the treatment. And with our real-time PCR analysis, we found the most changed pathway is PPAR and RXR alpha. Intact, and they are also, both of them are very responsive to the treatment. And PPAR and RXR together regulate the transcriptions of fatty acid metabolism genes. And so we've formed the hypothesis that retinoic acid, which is responsive to the NADH redox state, could stimulate RXR PPAR signaling pathways and regulate fatty acid oxidation gene expression. To see if this has a functional consequence in our model, we see that in the TAC heart, there's a significant downregulation of fatty acid oxidation for ATP production. And here, the white portion of the bar, and this can be restored when we treat the animals with NAD precursors. So this is all good, but we need to demonstrate that this mechanism we propose through the retinoic acid and PPAR RXR mechanism is sufficient to account for the benefit we see here. So we used a GW compound to inhibit this complex function. So here we used the isoproterenol-induced cardiomyopathy model, and without GW compound, we see a clear benefit in heart function in both knockout and wild-type animals with the NR treatment. With the compound on board, even though it didn't exacerbate the untreated animals, but it totally eliminated the benefit of the treatment. And we used a different model, a TAC model, to see if this is applicable for treating the patient who has already developed cardiac dysfunction. So here we induced TAC, and we waited for months until the cardiac dysfunction has already developed, and then we start treatment for another 2 months. Here we could see that if we leave the animals untreated, and the cardiac dysfunction will progress. If we treat it, we'll see improvement in cardiac function. So this is all very exciting, but they are all in mice. So to see if these findings are translatable, we collaborated with heart failure physicians, Kevin O'Brien at our institute, and we initiated a safety and tolerability study of NR in heart failure patients. So this is a stage C ambulatory heart failure patient. We did a 1-to-2 randomization to placebo or NR, and went through a dose titration to up to 2 grams per day and treated patient for 12 weeks. And so the result has already been published, and it's very well tolerated, and the NR indeed increased the whole blood NAD levels. And it's a very small study, so it's not powered to determine whether there's any efficacy on heart failure. But our preliminary study for this project showed some very interesting results using the peripheral blood mononucleated cells, PBMCs. So here just shows the treatment is very small number of patients for preliminary result. The whole blood NAD level increased. The PBMC's mitochondrial function improved, and here showing as the respiratory function, both at baseline and the maximum level, increased significantly. And this is associated with a decreased gene expression for inflammatory response in the PBMCs. And so this is unexpected results that we were mostly look at the cardiomyocyte function, but the PBMCs tell us that mitochondrial function appears to be important for the inflammatory response in heart failure. So follow up this study, we initiated heart failure study 2 that we're taking LVAD candidate, 3-to-1 randomization to unarmed placebo, and we treat the patient before the LVAD implantation. And so when during the implantation, we can get a piece of heart tissue from those patients. So the goal here is to ask very specifically, does the NR regime would increase myocardial NAD levels? And this has never been done by any study so far in human heart. And the other question is, does the change we see in the myocardium actually correlate with changes in the peripheral? And this would be very significant for clinical applications if we go forward. And of course, we're interested in the biological effect. And so this study has just finished enrollment, and hopefully we'll get data analysis done very soon. Now, last few minutes, I want to turn to the heart failure with preserved ejection fraction, HFpEF. And as all you know, now we understand that the heart failure comes in 2 different forms. The HFrEF with impaired ejection fraction, the heart basically cannot pump, and the HFpEF with preserved ejection fraction, the heart basically the blood doesn't return to the heart. And these 2 types of heart failure has somewhat different etiology, and they're both deadly, especially HFpEF that has a very limited treatment options, and we do not fully understand the mechanism. But if you look at the etiology of HFpEF, and the metabolic arrangement is derangement is very common in this group of patients, and suggesting the metabolism could play a role. And so we initiated a study with this Akira and Tim in the lab, comparing HFpEF and HFrEF, look at their metabolism and mitochondrial function. And so we're using TAC as a model for HFrEF, and we're using the 2-head high-fat diet and L-name as a model for HFpEF. And so this is just to see that we're successful in induce HFpEF and HFrEF in the mouse models. And here we looked at the cardiac energetics in the 2 models. As I mentioned, HFrEF has a very severe impairment of myocardial energetics. And it turned out that HFpEF patient also has a downregulation of PCR, as shown here. And these features are captured by our mouse models that both HFrEF and HFpEF has decreased PCR to ATP ratio. And so note here that the one-hit control, either the L-name or high-fat diet for the HFpEF model does not have the impairment of PCR to ATP ratio. And it's very well known that heart failure with reduced ejection fraction has metabolic remodeling. For example, the contribution of fatty acid for ATP production has decreased, and the alternative substrate, carbohydrate ketone, has increased its contribution. But in our metabolic HFpEF model, the remodeling is totally different, and it's the heart is highly dependent on fatty acid for energy production, and this is likely due to the high-fat diet component that, because of time restriction, I'm not going to show you the comparison of this profile to high-fat diet profile. It's very similar. But curiously, high-fat diet in our study does not impair bioenergetics, but this one has impaired bioenergetics. So looking into the underlying mechanism, we measured mitochondrial respiration using fatty acid as substrate isocarnitine. Here we could see that high-fat diet has a very well-maintained mitochondrial respiratory function for the duration of our model, but the HFpEF has a decreased mitochondrial respiration. This is due to in the high-fat diet, there's an upregulation of mitochondrial quality control, showing here increased mitophagy activity using mitochondrial tagged chyma as a reporter of mitophagy activity. And the similar findings if we use the LC3 levels when we block the lysosome activity. But there is no response in mitophagy in the HFpEF heart, even there both high-fat diet and HFpEF are obese and metabolically stressed, and the HFpEF has no response in upregulation of mitochondrial quality control. So this is very curious to us. Why does that happen? We performed lipidomics of the control heart and HFpEF and the high-fat diet animals, and you can see that both HFpEF and high-fat diet animals has very different lipid content in the heart compared to the chow. But these two also have somewhat slightly difference in some metabolites. And so when we looked at the differential level of the metabolites, and there's very few individual metabolites that is substantially changed with statistic significance. But you look at the whole group as a whole, there's more metabolites that upregulated in the HFpEF compared to high-fat diet, especially free fatty acid that's shown in red dots. All the free fatty acid this metabolomics program can detect on the increased side in the HFpEF group. So we wonder if there's something changed in the fatty acid oxidation pathway that fatty acid will accumulate in the heart. So this is a gene expression looking at the fatty acid, the key proteins involved in fatty acid oxidation. You can see in HFpEF, in high-fat diet, that they're all relatively upregulated, but in the HFpEF, they're showing red, there's no change at all. Especially CPT1, which is the mechanism for fatty acid to enter into mitochondria for oxidation. It's actually decreased in the HFpEF heart. So we ask whether the fatty acid oxidation are involved in this no response to mitochondrial quality control observation. So we use the iPS-derived cardiac myocytes that carries the chyma as a reporter for mitophagy activity, and we treat those hearts with palmitate to give it fatty acid. Here we could see a lot of orange dots here, and that indicate mitophagy activity has increased. And if we, at the same time, when palmitate is still on board, add atomoxia, which will inhibit fatty acid oxidation in the mitochondria, and this upregulation of mitophagy goes away. So we actually tried several fatty acids to get similar results. Suggesting it's not the fatty acid, but it's the oxidation of fatty acid is required to upregulate the mitophagy activity. So here, just to see if the lack of activation of mitophagy is really responsible for the phenotype that we see in the HFpEF heart, we try to manipulate mitophagy activity. One thing we did is we used the TAT-Becklin-1 peptide, which will mobilize more Becklin-1 and will form more autophagosome or autophagolysosome and promote mitophagy activity. And this indeed has improved mitophagy activities in HFpEF heart. The other thing we did based on our findings of fatty acid, we tried to stimulate fatty acid oxidation by delete ACC2 in the HFpEF heart. And here you can see by deleting ACC2, we're able to increase fatty acid oxidation both in the chow heart and in the HFpEF heart. And this is also lead to increased mitophagy activity in the HFpEF heart. And these measures, I want to show at the end that has clearly a functional benefit here. So all these animals I show here are HFpEF animals, but the open bars are control animals, such as flox animals or Cree animals, and these are the knockout animals. And so the knockout animals has reduced cardiac hypertrophy, reduced pulmonary congestion, as well as improved diastolic function. The systolic function doesn't change, and the myocardial energetics has improved, and the oxygen, the mitochondrial respiration has also improved. So I want to summarize this up, that the data I showed today shows that both HFrEF and HFpEF heart presents impaired energy homeostasis and mitochondrial dysfunction. And there are multiple mechanisms that could contribute to mitochondrial deficiency in the failing hearts. And the result I shared today suggesting there are roles for mitochondrial biogenesis, fatty acid oxidation gene expression, and for quality controls. Different type of heart failure might involve different mechanisms, and but regardless of that, and our initial results show that strategies that targeting mitochondria, either by change NAD/NADH redox or fatty acid oxidation pathways, has benefit in animal models, and it has also shown promise for heart failure therapy in human. So I will end here, but not last but not least, to thank many people who has contributed to the study. The NAD/NADH study was led by Matthew, who is now our junior faculty member, and carried on the MRPP2 directions. And Hongye was a student who worked with Matthew. And Dennis and Bo did the PBMC studies and clinical trials of NR studies, and this is now Dennis' direction, looking at the PBMC and the inflammation. And Akira and the team did HFpEF studies, and Tim has returned to Japan and becoming a physician scientist, and the lab continue to work on HFpEF and mitochondrial dysfunction. And everyone in the group has contributed to the discussions and to the wonderful environment that I'm really lucky to work with in the past many years. And the collaborators, I already mentioned Nina, who did the retinoid and acid metabolism, and the Raftery group did our metabolomics analysis. There's a very special thanks to New Sun, who provided us a mitochondria-tagged animal models and iPS cells. Thank you very much, and I'm happy to answer questions.
Thank you, Dr. Tian, for a really interesting talk. Both of us were here in the studio, madly scribbling notes during your talk, so I think there'll be lots of questions from us and also from our audience. Just to organize the questions a little bit, our discussion, I thought we might start first with clinical implications, and then talk a little bit about the animal model, and then really dive deeply into the mitochondria. And maybe we'll even get into one of your favorite topics, mitochondria, metabolites, and epigenetics at the end. I was really interested in this trial that you're doing. You are giving nicotinamide riboside, NR, to patients with heart failure with preserved ejection fraction. Is that right? Or was that HFpEF patients?
What we've done so far are reduced ejection fraction.
Reduced.
And Yeah, I think there are talks of doing it in the preserved ejection fraction, but our group hasn't done that yet.
Yeah. Well, so it was HFrEF, heart failure with the reduced ejection fraction. And you found that 2 grams of NR, nicotinamide riboside, 2 grams a day, had an effect on your patients. You measured blood cytokines, inflammatory cytokines went down. And also there's some evidence that aerobic capacity increased with that administration. I think this is a supplement that's available in the stores, right, NR?
Yeah, NI is a nutritional supplement. I think when we start this study, we thought that use a nutritional supplement would be the quickest way to get to the human studies, because you don't need a lot of you don't need to go through a lot of regulatory steps. But the dose we gave is much higher than recommended nutritional supplement dose. Mm-hmm. We also have some separate studies looking at the pharmacokinetics and how much you need to take and to increase the levels of NAD. And so far, most of the studies taking this approach, giving the precursors of NAD, are measuring the NAD level in the blood, basically the level in the blood cells. And there are very few who are able to measure it in the tissue. I think the challenge is to see whether it has increased the level in the tissue.
Mm-hmm. It may be too early to answer this question, but any thoughts about the safety of the supplement?
So the study we published a couple of years ago did a large panel of the blood test and the follow up, the compliance of the study. So 2 grams for 12 weeks, we did not notice any adverse event. So the study 2 that we gave it to 5 to 10 days to LVAD patient, those are very, very sick patients. We gave it only for a short period of time, and I believe we did not receive any report of adverse effect.
Okay, thanks. Well, that's promising. Look forward to seeing more data on that clinical trial. Did you have a question about the patients? You're going to hold your fire, keep your powder dry for the more basic stuff. I did have a question about the animal model that you're using. You use an animal model that's well established by Dr. Joe Hill's group. The high-fat diet plus L-name is the chemical that's used. L-name is an antagonist of NO synthase. So that does raise the question of what is the role of nitric oxide in heart failure with preserved ejection fraction? Because presumably that L-name by blocking nitric oxide is somehow accelerating HFpEF pathology. So tell us what your thoughts are about that. How is a deficit of nitric oxide, that endogenous vasodilator, how is that having an impact on HFpEF?
Yeah, that's a great question. I think when the study was first published, there are a lot of follow-up studies to look at the protein modifications related to nitrate metabolism, and there are I think there are pathways mostly on the ER stress pathways and the ribosome biosynthesis pathways were involved, and so this it has a very complex signaling pathways. For us, the L-name inhibit ENAS could be responsible for some metabolic changes that we see in this model, because we are also very intrigued that this model has a very dominant effect of high-fat diet obesity, but it doesn't act the same way as high-fat diet. So the inhibition of ENAS has been shown to change fatty acid metabolism. So maybe that is why these and the HFpEF animal models has a different response to fatty acid metabolism compared to high-fat diet only.
All right, so if I understood you, I mean, this effect of L-NAME blocking nitric oxide production could be beyond its effect on vascular function, vasodilation, and vascular structure, which it is, of course, known to affect. It could be due to a direct effect on the mitochondria. And my understanding is that nitric oxide itself does have an effect on mitochondrial activity. Is that what you're thinking?
Yes, and the L-name there was I think originally it was there to induce high blood pressure because that's a risk factor for HFpEF, but it does way more than that.
Yeah, I think that's right. Well, I'm going to turn to my colleague here in the studio. I know she's burning to ask a few questions about metabolism, her favorite topic.
Yes.
Go ahead, Lily.
Yes. So I think the second story you share with us is the role of mitophagy in HFpEF, and also you're focusing on the condition of obese condition, right? Since you're comparing high-fat diet versus HFpEF, right? So I just want to know if other forms of cardiac stress, like hypertension or inflammation or diabetes, can similarly impair mitochondrial pathway and also the mitophagy.
Yes, so I think HFpEF study is still struggling with the model because there's clearly a different model might have emphasized different risk factors. So the model that we're using, people usually call it metabolic HFpEF because obesity and high-fat diet is an important component. But you can also induce so-called hypertensive HFpEF. So it's mostly hypertension, renal dysfunction. It has a minimum component of obesity. So mechanisms could be somewhat different. And another important, for hypertensive model, another important component is aging. So people talked about that little old lady, and it's so the woman is not obese but old and hypertensive. So for mitophagy activity, aging is a very important factor in there. So mitophagy activity and mitochondrial quality control is impaired in aging.
Yeah. You just on that topic, mitochondrial damage is one of the hallmarks of aging, one of the processes involved in aging. And I wonder if you could talk a little bit about that, about mitochondrial DNA damage that occurs with aging and its influence on metabolism, and how is that repaired? How is mitochondrial DNA damage repaired? Is there some hope for us? Can we repair our mitochondrial DNA damage? How do we do that?
Yeah, that's a really important topic, and I think mitochondrial DNA damage is first of all it's quite severe and it's probably received less attention, and so mitochondria is the site of RAS production. It has enormous amount of RAS produced. So mitochondrial DNA is basically exposed to that. And the mitochondrial DNA is not very well protected. So it's also very vulnerable to RAS production. So the mitochondrial DNA mutation over time and over the life is accumulating. This is well known. And it's especially true in heart failure conditions. We have previously published studies showing that the oxidative mitochondrial DNA damage would actually impair its replication as well as transcription. So for mitochondrial DNA, the transcription and the replication are linked. They start from the same site with a similar mechanism. So I think the mitochondrial DNA damage is quite a significant role in regulating mitochondrial function in those disease states. Plus that, when you have a DNA damage and it triggers the repair, if it triggers repair process, and it will mobilize proteins, enzymes that could consume NAD, so which could also impair the NAD/NADH balance in that way.
You just mentioned interaction between the nucleus and the mitochondria and DNA, mitochondrial DNA repair. My understanding is that telomerase actually can migrate to the mitochondria and plays some role in mitochondrial DNA repair. Any thoughts about how telomerase might be doing that, or what are the other mechanisms of mitochondrial DNA repair?
Yeah, that is a really intriguing mechanism, but we haven't looked into those, and maybe some other studies have shown that, and we should look into that in future studies.
Yeah. The mitochondria in the cell, are they homologous? I mean, are the DNA in those mitochondria, my understanding is it's maternal. It's from the mom, the mitochondrial DNA. Is it all the same, or are there SNPs between the different mitochondria? Do they have differences in the mitochondrial genome?
Yeah, that's a great question, and so it is not homogeneous, and there's for the nuclear DNA, one cell has one copy, or maybe you have two nucleus. That's all. But there's hundreds and thousands of mitochondria unit in the cell, and they could have different. Slightly different version of mitochondrial DNA, even though this mitochondrial dynamics and mix and match of different units and select the ones that's more damaged to mitophagy would get rid of some of those unhealthy copies, but it doesn't really make it completely uniform. And because there are so many units of mitochondria, and this concept is also there, that is not all the mitochondria are functioning at all times. So when the cell so this discussion has always coming up. If the cell performs a high function, maybe all mitochondria are in and perform 100% of its capacity. But when it's at a baseline function, do you have every mitochondria perform at 50%, or only 50% of mitochondria perform at 100% capacity? So these are not really known. I think our science technology has got to the single cell level, but we haven't got to the single subcellular level yet.
So I was very interested in your previous slides showing the differential substrate preference in different types of heart failure. So while you're talking, your talk is more focused on fatty acid oxidation. I also noticed some other major metabolites change a lot. Like for example, the lactate is almost doubled. The preference is almost doubled in the HFrEF, and also cut it into half in HFpEF. So I was interested in what do you think the role of lactate in the heart failure, and if there is any differential role in different types of heart failure?
Yeah, great question. I think there's a lot of interest in lactate now, and in the HFrEF, clearly both in animal models and our study and others, and as well as human studies recently done, looked at, see that the contribution of lactate for ATP production is quite high in the heart failure compared to non-failing status. And it is still a question of lactate use is increased. Is it because the fatty acid use is decreased? So lactate is just here to filling the gap, or is it there's active mechanism to push lactate in? I think that is still under investigation. In the HFpEF study. Because ours is a obesity HFpEF study, I think the lactate downregulation for oxidation is mostly because the fatty acid is quite dominant in that way. So both lactate and the ketone contribution decrease. So these are all relative contributions. It's not an absolute rate. It's just relatively it uses less. But it doesn't mean the lactate level in the cell is lower. What we're measuring is only the lactate that's being oxidized to produce ATP. That use is decreased.
Okay, so I think I have a following question is what do you think the role of metabolites in epigenetic regulation? So a lot of metabolites are known to serve as an epigenetic regulator. So I know you talk a lot about the bioenergetics, but do you also work in the crosstalk between metabolites and epigenetics?
Yeah, fantastic question. I think that's a huge field that is wide open for us to look into. And so metabolites definitely modify. It modifies genome, it modifies proteins, and there's more and more of them identified. So originally, the focus is mostly on acetylation or methylation, but now there's many other modifications, palmitoylation, lactylation, and so that involves lactate, and they all have very significant role. And I think that field is wide open, and with more advancing technology, the better we can look into this, and we will know more.
Yeah, I think it's really interesting, right? I mean, we used to think that the glycolytic shift in heart failure was, well, it's just a different fuel source for the heart. But Lily in our department and others have shown that in the vasculature, that glycolytic shift plays a really important role in cell fate transition. So we have evidence to believe that this glycolytic shift in the vasculature is maybe playing a role in some of the pathophysiology that we see, the loss of vascularity and the increase in fibrosis. That might be due to cell fate transitions, specifically endothelium to mesenchyme transition may be driven in part by that glycolytic shift and its effect on epigenetics. And I wonder if you have any thoughts about that.
Totally. I think there's so much talk about metabolism beyond ATP production. So it's the pathways that was very difficult to measure previously, and because the ATP production pathway is so robust and so many ways to measure it, but those pathways are difficult to measure. But pathways in glucose metabolism, for example, are very, very closely linked to cell growth and proliferation and cell fate determination. So that's actually unique for glucose metabolism. Other substrates, such as fatty acid or ketone, they enter directly into the TCA cycle. So they missed all these pathways above the TCA cycle, the intermediate pathways. So the pentose phosphate pathways, nucleotide biosynthesis, and so those are mostly filled by glucose metabolism.
Well, Dr. Tian, we've reached the top of our hour today. I want to thank you from myself and Lily on behalf of our crew here at Houston Methodist for joining us today and talking to us about metabolism and heart failure. Just a wonderful talk. We're going to follow your work. We look forward to the clinical trial that's underway, and best of luck with that. Thanks again for joining us at the cutting edge of cardiovascular sciences, Dr. Tian.
Thank you.
Thank you. My pleasure. Thank you for having me.
We're clear.
All right.
So